生命科学分野でジャパンプライズを受賞されましたエマニュエル・シャルパンティエ博士とジェニファー・ダウドナ博士にお話をいただきます。
It's for sure a great pleasure for me to be here, and my colleague Jennifer Doudna and myself, we are very humble and very pleased to be recipients of the 2017 JapanPrize. This is a great honor. So I very much enjoyed the talk of Dr. Shami, and I think there are some correlations with cryptography and CRISPR-Cas9. It's about breaking the code. It's also about infections. I think CRISPR-Cas9 has infected the scientific community in the sense that the scientific community has really broadly adopted this technology. But it's also about infections happening in bacteria. And there is also privacy, because biologists like to know everything about genomes and their privacy, their secrets. So I find this very interesting in terms of correlations. So hopefully, Jennifer and I, we are not going to be too much overlapping. We have prepared our talks to not overlap too much. I would like to start by showing you really what the CRISPR-Cas9 technology is about. So I will give you more insights, but in principle, this is originally a system that exists in bacteria and that allows bacteria to fight viruses and other types of mobile genetic elements infecting the bacteria. It's also existing in archaea. And these systems, CRISPR-Cas, existing in bacteria, in the case of CRISPR-Cas9, has been harnessed in a very powerful technology that allows to target DNA of any cell and organism, and allows, in terms of the CRISPR-Cas9 technology, to really modify sequences of DNA in a very precise fashion and in a very efficient fashion. So here you have the DNA to be targeted by the system, and here the system is composed of a protein component, here represented in blue with the form of scissors, and this protein is called Cas9. The protein has enzymatic activity and is guided by an RNA component that is in nature a duplex of two RNAs, the tracer RNA and the CRISPR RNA. And this RNA molecule provides the system its programmability. So this RNA molecule works like a kind of GPS system, allowing the enzyme to recognize sequence-specifically DNA and to cleave the DNA with surely the need of a presence of a sequence on the DNA for the system to work. So in principle, it is a very simple system to use, and the very nice aspect of the technology is that it's programmable. But what is important to say is that the enzyme will cleave the DNA, and the rest is done by the cells. And depending on how the technology is programmed and is designed, different modifications on the DNA can be made. And so this field of research, the usage of the technology CRISPR-Cas9 started in early 2013 and was already highlighted in the magazine Science nearly 4 years ago as the CRISPR craze, with the subtitle, "A Bacterial Immune System Yields a Potentially Revolutionary Genome Editing Technique." And this is really about this technology that allows precise gene surgery in any cell and organism. And this paper was published just a couple of months after a number of publications came out in scientific journals at the beginning of 2013, showing that the CRISPR-Cas9 technology was working very well. And this was only a couple of months after Jennifer Doudna and myself, we published a paper in Science in the summer 2012, describing the last step of the CRISPR-Cas9 mechanism in bacteria, so the mechanism behind this last step of the immune system, and describing how biologist could really design the technology for the purpose of precise genetics. So this CRISPR-Cas9 technology has been the highlight of a number of scientific journals over the last 5 years now, nearly 5 years, was highlighted at some point as a next medical breakthrough, and it has made also the highlight of a number of journals for the public. And so it has been quite much in the media, and it is important for us scientists because it really highlights really a mechanism, a basic mechanism of life, which we have studied, interested in understanding fundamental aspects of life, and really a basic science leading to this powerful technology. Now, the CRISPR-Cas9 for my case came really from my interest in understanding bacterial infectious diseases, and a lot of colleagues of ours are interested in understanding mechanisms of life in different organisms and models. Some are really interested in disease affecting the cancer, heart, genetic, or genetic diseases, or diseases affecting the brain, or other types of infectious diseases also involving viruses, fungi, or parasites. And we are mainly interested in understanding the mechanisms, explaining the development of diseases, and always with the hope to identify pathways, genes, and molecules that could lead to the development of novel therapies and novel technologies. And this is really what CRISPR-Cas9 comes from, and CRISPR-Cas9 now is very useful as a genetic tool to understand better this phenomena. With regard to infectious disease, I just want to point out here that this is a leading cause of death in children and adolescents worldwide. There is still an antibacterial resistance that poses a major health threat. And at least with regard to CRISPR-Cas9, like for other types of disease, it's really a tool that will really increase tremendously the knowledge of interaction of bacteria with the human host. Having said this, genetics is really important to, as a technology to understand the functions of genes. And here I would like just to bring some aspects of the history of genetics that really started in the 19th century with various rules with regard to fundamental genetics that were established by Darwin on the origin of species, Mendel on the segregation of alleles, and in 1871, Friedrich Michel who isolated DNA. Then the 20th century, the mid of the 20th century, was very important with regard to the information. So showing that the DNA was the carrier of genetic information, the structure of the double helix of DNA that was determined, and the genetic code that was revealed. And surely the need to understand the genetic code and unravel the functions of the genes. A number of interesting molecular biology tools emerged starting at the end 60s and mid 70s. And what I would like to mention with this regard is that most of the enzymes that have been used as molecular biology technologies originate from bacteria or viruses, or trying to understand how bacteria defend themselves against viruses. And with this regard, I like very much the quote of a French microbiologist, François Jacob, who was quoting, "Nature is a tinkerer, not an inventor." And it's really all about how to tinker DNA. And this is what a number of viral and bacterial enzymes know very well how to do. So this field of research, understanding the enzymes and other important molecules for molecular biology purposes have been so very important for the basics of the genetic tools in the mid-70s, to also allow to sequence the DNA, to amplify the DNA. And then a field of research and application that started in the 90s, with two types of nucleases, zinc finger nucleases and talon nucleases, that were very useful for precise gene editing. And these nucleases work differently than the CRISPR-Cas9 system. They work nicely. However, their mechanisms did not allow to really lead to a democratization of these technologies. And this is really what CRISPR-Cas9 brought, the possibility to introduce changes on the genome in a less time-consuming manner and in an easier fashion. So for my part, I would like to mention that CRISPR-Cas9 originates from the interest of my lab in working with Streptococcus pyogenes. And actually, the system that is used worldwide really originates from this bacterium, Streptococcus pyogenes, a human pathogen, so responsible of a large range of diseases in humans. Here you see the Streptococci colored in red for the purpose of the picture, invading human epithelial cells colored in green for the purpose of the picture. We are mainly interested in understanding how bacteria attack the human host and how the human host defends itself against the infections with bacteria. But you have also another kind of infection biology where the bacteria are considered as hosts, and they can be attacked by mobile genetic elements such as phages, the viruses of bacteria, or plasmids, so these are autonomous DNA molecules that can bring new genes to the bacteria that can be beneficial for the bacteria or detrimental to the bacteria. And surely, as we developed an immunity against bacteria and viruses, bacteria as hosts have also developed immunity systems to be able to deal with infections with mobile genetic elements. And here I would like to show you a phage, so a virus, infecting a bacterial cell, whereby the DNA of the phage is injected into the bacterial cell. In this case, when the phages, the viruses of bacteria, enter the lytic cycle, the DNA of the phage will replicate, and there will be production of viral particles that can kill a bacterial cell and propagate to kill other bacterial cells. So surely here it is important for the bacteria to defend themselves against death. And you have the so-called temperate phages that can enter the lysogenic cycle, whereby the invading DNA of the phage will integrate into the genome of the bacterium, and this will allow to bring new genes, new characteristics to the bacterium. And those prophage DNAs can be transferred through cell division to the further generations. And here again, it is important for the bacteria to be able to deal with maybe some DNA elements which are unwanted and to be able to fight this infection with temperate phages. And this is where CRISPR-Cas enters into play. There are different immune systems which bacteria have evolved, and CRISPR-Cas is actually considered as the first and only so-called adaptive immune system. So it works as follows, very roughly. The system is composed of a series of genes that encode the protein components of the system, and a so-called array that encodes the RNA components of the system. So you have a protein components and you have the RNA components. And you have first a phase where the phage infection is going to be memorized by the system, whereby some CRISPR-associated proteins encoded by the CRISPR-associated genes will recognize the genome of the phage while entering into the cell, will insert a portion of the phage DNA into the so-called array, and this will work as memorization marks. So in principle, all these sequences of colors on the genome correspond to different infections with different types of phages, plasmids, or other types of mobile genetic elements. So this is the way the bacteria can memorize the infection. And then there will be the phase of destruction of this here, in this case, virus, upon a second infection. And in this case, the RNA components are, for this step, are transcribed and translated. The RNA component of the system is transcribed and undergoes what we call processing to lead to the production of unique CRISPR RNAs that each are composed of a specific mark corresponding of a certain mobile genetic element. And these individual CRISPR RNAs will guide a complex of CRISPR-associated protein to the invading genome upon a second infection of the phage. And via this sequence of the RNA molecule that corresponds to the memorized mark of the phage DNA, and by base pair complementarity with the genome of the newly infecting phage, one CRISPR-associated protein will be able to recognize this interaction and cleave the genome of the virus. So you have a very complex history with regard to the CRISPR-Cas field. I just want to mention that it started in 1987 with the description of the repeats of the system by a Japanese group. And since 1987, nothing really happened very much after 15 years. But since the beginning of 2000s, a number of colleagues of ours have understood that the system is an adaptive immune system existing in bacteria and archaea, and have deciphered different types of CRISPR-Cas mechanisms. Now, from our part, we focused on the CRISPR-Cas9 mechanism, that is one of the CRISPR-Cas systems existing in bacteria, and using, I will say, a very precise genetic approach in Streptococcus pyogenes, as well as a biochemical approach, we identified the components of the CRISPR-Cas9 system with regard to the phases that are important to destroy the virus upon a second infection, composed of this enzyme Cas9 and two RNA molecules, the tracer RNA molecule and the CRISPR RNA molecule. So we really started from the identification of tracer RNA. Cas9 and CRISPR RNAs were already identified, and worked through the pathway to really understand that the system is composed of one enzyme with two RNA molecules forming a duplex and allowing to bring Cas9 to the right sequence on the invading genome of a virus infecting the bacterium. And in this case, here there are two scissors. The enzyme, guided and programmed by this duplex of RNA, can cleave sequence-specifically the DNA, and you have a break in the target DNA. And this can be exploited to really allow diverse modifications on the DNA in a specific manner at a specific site on the genome. I just would like to mention that the system used by the scientific community mostly is a system from Streptococcus pyogenes, but these systems in bacteria have largely evolved, and in the case of CRISPR-Cas9, it has evolved to really be present in more than 350 bacterial species, having some diversity in terms of sequences that are the composition of the Cas9 protein and the RNA molecules. However, the mechanism is globally similar, even though you have differences in terms of specificity and efficiency. And I just wanted to mention this because it's a very important aspect of the CRISPR-Cas research. Is really the evolving system that is extremely high for this immunity. So, and I would also like to mention that the main work on CRISPR-Cas9 has really been done on Streptococcus pyogenes and using the Strep pyogenes system as a technology. And now there are some efforts to try to diversify the CRISPR-Cas9 system based on the knowledge that we can see in nature. So after we really understood the CRISPR-Cas9 mechanism, how it can assemble together and how it can target DNA. So this, for the second step of the mechanism, it was published in Science in 2012. We already emitted the hypothesis at that time that RNA programmable Cas9 would offer considerable potential for biotechnology call biomedical and gene therapeutic applications, that it would offer a number of advantages compared to the technologies that were used prior to CRISPR-Cas9, like zinc finger nucleases or TALEN nucleases, that the technology would offer the possibility to have a system cheap and easy to design and use, the system being efficient, the system being also very versatile. Not only you can add new sequences at a certain point of interest on the DNA, but you can also replace sequences by new sequences. You can specifically correct mutations or introduce mutations. You can modify the DNA. You can also label the DNA. You can use a technology as a multiplexing approach, meaning that you can design the tool to recognize different sequences of DNA at once. So you can perform different modifications within one step. And as I said, you can really design the tool into different various flavors. The system is working very well and is very efficient. There are a lot of efforts from the scientific community to improve the specificity of the technology and try to reduce what we call the off-target effects, meaning making sure that the scissors are going to recognize a certain sequence of interest and not another sequence on the genomes. And you have a lot of efforts that have led to very interesting insights in this direction. And the challenge that, the main challenge that remains, that is to bring the technology to cells and organisms, whereby here delivery technologies are very important with regard to bringing CRISPR-Cas9 to the right type of cells and tissues or organisms. So a lot of efforts in the scientific community. Now that there is really a tool that can target very well the DNA, delivery technologies are very much developed. So as I said, you can correct sequences of DNA, delete sequences of DNA. You can add new sequences at a specific spot on the genome. You can replace sequences by other sequences. The technology has been shown to work very well in plants, in human cells, in animals, different kinds of model organisms, in fungi, and also in bacteria and archaea. But it's a very, very useful tool to understand the biology of eukaryotic systems, including plants and human cells. It has very broad applications in biology, and I'm sure Jennifer will mention this point. I just would like to mention three aspects. It's very useful in synthetic biology to engineer new pathways, so it's very, very useful for biotechnological purposes. It's also very useful for agriculture, for the production of new types of crops, and in human medicine on different aspects. It is surely, with regard to research and development, a technology that is really powerful to understand functions of genes and unravel new pathways in a large variety of cells and tissues. And this is important to mention because now one has a tool that allows to really accelerate the understanding of biological functions in a large diversity of primary cells and other types of cells. It's very useful to engineer new disease models, including humanized models, to really screen for new targets for therapeutics with regard to development, to validate new therapeutic targets and medicine that are developed, and also very useful for the bed-to-bench approach with regard to the efforts to get the knowledge of sequences of genomes of patients where genetic predispositions for diseases are hypothesized, and where one can go back to the bench and test possible mutation linked to a certain diseases. And surely a very interesting aspect that is to develop the technology as a direct gene medicine in the aspect of treating certain human genetic disorders. I just would like to finish by saying that the technology is also very useful in the field of bacterial infectious diseases, specifically when one focuses on human diseases, and when animal models have been majorly used. specifically those models that are not always the best models to use when one is really working on a human bacterial pathogen like Streptococcus pyogenes that infects only human hosts. So now we really have the possibility to have a very powerful genetic tool that allows to modify human cells, also create these humanized models allowing to understand better the interaction between bacteria and the human host. And I just would like to finish by saying that my laboratory is focusing on not only on CRISPR-Cas, but also on other aspects of research involving proteins and small RNAs like CRISPR-Cas, but working differently, and having roles in the regulation of gene expression, allowing to understand how bacteria survive in their environment, how bacteria can produce virulence factors or other types of factors. And I just mentioned this because this research at the fundamental level has been the start for me for the CRISPR-Cas9 research. And I really hope that during the next years of my career, I will have the chance to highlight interesting mechanisms that could potentially lead to either new technologies to target genomes or new anti-infective strategies. Brief acknowledgements from my side saying that this research on CRISPR-Cas9 started at the University of Vienna in Austria. The main work was done when I was in Sweden at the Laboratory for Molecular Infection Medicine Sweden at the University of Umeå. Then I moved a couple of years ago to the Helmholtz Center for Infection Research in Germany, and now I'm at the Max Planck Institute for Infection Biology. So I would like to thank those institutions and a number of fundings that have been very helpful for supporting the research on CRISPR-Cas9. I would like to thank a number of students and postdocs in my lab who have largely contributed to really the understanding of the CRISPR-Cas9 pathway, specifically Christoph Schilinski, Eliza Delcheva, also Ines Fonfara, and Maria Eckert at really the very beginning. The group of Jörg Vogel for the help in deciphering the step of the CRISPR RNA maturation. The group of Jennifer Doudna Schorley with Martin Jinek, who's now at the University of Zurich in Switzerland, for the step of DNA interference, and also collaboration with a group of Eugenie Kounin for the evolution aspect of the system and some bioinformatic studies. And this is the picture of my lab this summer when we had a number of summer students. And I would really like to warmly thank my team for all the support that the PhDs, postdocs, and other members of my lab can provide me. And I would like to thank you for your attention. And just to finish, I need to mention for a conflict of interest purposes, ERS Genomics and CRISPR Therapeutics, which I have co-founded with Rodger Novak and Sean Foy. And I would like to thank you for your attention. And I will be happy to give the stage to Jennifer Doudna, my colleague. Thank you very much.
Okay, well, good evening, everyone. It's an incredible pleasure to be here. It's been a really interesting week so far, and I learned a lot already tonight about the science of our colleague, Dr. Shamir, fascinating lecture. And like Emmanuelle, I felt there was a lot of interesting parallels between your work in cryptography and what we've been working on with the information code in cells. So I'd like to basically continue the lecture that we just heard from Professor Charpentier by talking about a little bit more about the molecules involved in gene editing and where this technology is going. And I think one of the things that has been so interesting to us about this field is just how rapidly it's been developing. So you may have noticed that we published the paper together on the mechanism of this system in 2012. And here we are in 2017. And already there are clinical trials going on using this gene editing technology in human patients. I think that's kind of a remarkable thing. And so I'd like to just take a step back and reflect a little bit on the mechanism of this, which is sort of always been my interest in molecules and how they operate, and then give you a perspective on where the field is going, and I think what some of the interesting challenges are going forward, not only technically but also ethically, with a technology that's very powerful for controlling the genetic code in cells. So just to remind everyone, so you know that DNA is the molecule that holds the genetic information in cells. So instead of a computer code, we actually have a code that's made up of chemical letters. And in human cells, this is a code that's 3 billion base pairs or pairs of letters in length. So it's a very long code. And we've understood from the 1950s that the structure of DNA is a double helix, like shown here. And so already when scientists had first figured out this double helical structure, it was clear that this would explain how genetic information is stored in cells, how it can be faithfully copied, and not only that, how it might be altered in various ways. So I think that if we really go back to the beginning of genome editing, it really began with the understanding of the structure of this amazing molecule. And so with the understanding of the structure of DNA, scientists, if you look back over the last few decades, have been asking, how can we manipulate that information? How can we read it by sequencing it? How can we write it by synthesizing it? And then how can we rewrite it by changing it? And the CRISPR-Cas technology is really a technology that is analogous to a program on your computer that allows you to change letters or words or sentences in a document. So it's sort of saying, what if we had, what if a cell's DNA could actually be edited the way we edit the text of a document? And somebody made me this little this little widget that shows this. But the idea is that you could literally look at the code in a cell and say, I'd like to change that gene. I'd like to swap it out. I'd like to erase some of the code and replace it, or not. And I'd like to do it at exactly the site or position that I'm interested in altering and nowhere else. And up until recently, that kind of technology was really sort of out of reach. And the work that I did in my lab at UC Berkeley with the lab of Emmanuelle Charpentier started off with a very different question. It was namely the question of how bacteria fight viral infection. So this is showing you the surface of a bacterial cell with viruses landing on the surface. And these cells then have to figure out a way to defend themselves from the DNA that's getting injected into the cell. And so we started off asking that question, but as you heard from Professor Charpentier, the project went in a very different direction in the end, because once we understood the molecular basis for the CRISPR immune system in bacteria, it was immediately clear that this could be harnessed for a very different purpose, namely as a gene editor. So to explain that, there's sort of 3 things I want to touch on over the next few minutes. I want to talk about a little bit about the biology, and in particular about the mechanism of this pathway. I wanted to explain sort of the link between understanding the fundamental mechanism of this pathway and how it became a powerful technology. And then I want to turn towards the end to some of the ethical challenges that we now face, we being human society, faced with the possession of a powerful tool that offers enormous opportunities, but also the potential for misuse or even unintended consequences. Okay, but first the biology. So when a cell is infected by a virus, so here I'm showing you a cartoon image that shows a virus landing on the surface of a bacterial cell. And so the DNA of this virus is packaged here. It's under incredibly high pressure. And so when this virus lands on the surface of a cell, it injects the DNA at very high velocity into the cell. So you can see some of the DNA going into the cell. Now this DNA of the virus encodes all of the proteins and other molecules necessary to make more virus. So the cell has very little time, once it's infected like this, to defend itself before the cell will be taken over by these invaders, and many more of the viruses will be made, and the cell will be destroyed. And so that's where CRISPR comes in. And so you heard from Emmanuelle a nice explanation of this. And basically, the word CRISPR is an acronym that stands for clusters of regularly interspaced short palindromic repeats. Can't say that 5 times fast. And it refers to a set of sequences in the bacterial genome, this circular molecule right here, that was actually first noticed by a Japanese group and published in a paper in 1987. At the time, nobody knew the function of these. But what was interesting was that it was noticed in sort of the mid-2000s that this series of sequences contains little pieces of viral DNA sequence shown by these colored boxes. And they're placed in this array in a very particular way, so that there's an identical sequence shown by the black diamond that is repeated over and over, and in between are the sequences coming from viruses. Also, right next door in the genome are CRISPR-associated or Cas genes. These are genes that encode proteins that we now know are part of this system for defense against viruses. So let me show you a brief video that will summarize a lot of what you heard in Emmanuelle's talk and explains how this CRISPR system works as a viral defense mechanism, and also how it can be employed as a technology.
Bacteria use an adaptive immune system called CRISPR to acquire protection from viruses. When a virus injects its DNA into a bacterial cell during infection, a piece of the viral DNA is integrated into the bacterial genome, its genetic program, at a site called CRISPR that includes a series of repeated sequences that flank each inserted piece of viral DNA. This creates a permanent genetic record of past infections. Then, the sequences are copied into strands of RNA, DNA's chemical cousin, and the RNA is chopped into functional units, each containing one viral sequence plus the flanking repeat. The chopping process requires a separate RNA called tracer RNA that together bind to the Cas9 protein, a component of CRISPR immunity, to form a surveillance complex. In the laboratory, the functional parts of the two RNAs can be combined into a single guide RNA that holds onto Cas9 and programs it to find and cut a desired DNA sequence. Upon finding DNA with a matching sequence, the Cas9 RNA machine holds onto the matching DNA sequence, unwinds it, and cuts each strand of the DNA double helix. In bacteria, the broken viral DNA is chopped up and destroyed by other proteins, thereby halting the infection. But in animal and plant cells, this kind of RNA-programmed cutting triggers cells to repair the broken DNA by making small changes at the site of the break. In this way, a bacterial immune system was harnessed as a powerful technology for site-specific genome editing.
So you might wonder, how did somebody like me, who's always had an interest in molecular mechanism, and I've studied over the course of my career so far the way that the genetic information in cells is controlled and regulated, how did I get involved in studying this pathway? And to explain that, I just take a brief step back to what we are taught in, I guess, most high school biology classes these days about something called the, flow of genetic information in cells. And what this shows in this cartoon is that DNA encodes the information to make a cell or a whole organism, and in many places in the DNA, this molecule is copied by a process called transcription into RNA. And if that RNA is a messenger RNA, then that means that it's the messenger that encodes a sequence of a protein. And by a process called translation, the cell can read that code in the RNA and produce a protein. And the protein might have the ability to copy DNA or do all of the other jobs that have to occur in the cell to allow it to grow and function. But in my career, I've always been interested in the part of the pathway that stops at the level of RNA, because what was recognized back in the 1980s was that RNA molecules don't always encode proteins. Sometimes they have functions on their own in cells. And in fact, many scientists now think that RNA molecules explain the origins of life on our planet, that RNA molecules originally had the ability to both encode genetic information and make copies of that genetic information and pass it on to future generations. So when I was in graduate school, I got very interested in that process, and really over my whole career, I've been studying different kinds of RNA molecules to figure out how they work and how they control the flow of genetic information. So in the CRISPR pathway, it turns out, so here I'm showing you a cartoon of this pathway. So here we're looking at a bacterial cell, part of a cell. So this is the cell membrane. And you can see a virus infecting this cell and injecting its DNA into the cell. If that cell has a CRISPR system in the genome, then it has the ability to integrate little pieces of the DNA from the virus into the CRISPR sequence and store a genetic memory of that infection for future use. Now the key is, how does that information, how does that stored information protect the cell from future infections? And the way it works, and this is where my interest comes in, is that the cell makes a little copy of that genetic information in the form of an RNA molecule. And the RNA is then subsequently chopped into smaller units, each containing a sequence that comes from one of those viral infections. Those RNA molecules then assemble with one or more proteins encoded by the Cas genes to form a protein-RNA interaction that can use the genetic information in the form of RNA to search through the cell to find a matching sequence in DNA. And if that sequence match is found, then the proteins that come along for as part of this complex are able to destroy the viral DNA. And so in 2011, so my lab we had started studying this process back in around 2007 or so, and we're doing a number of experiments, biochemical experiments and structural biology to understand basically this part of the pathway right here, how the RNA was made and how it assembles into these complexes. But I knew that this was in many ways the key to the way the system worked, was how these targeting complexes recognize and cut DNA. And that's where my introduction to Emmanuelle Charpentier at a meeting in 2011 comes in, because we got together and realized that there was an exciting opportunity to work together to figure out the molecular basis for one type of CRISPR pathway in which a single protein called Cas9 was predicted or sort of hypothesized to be involved in viral DNA detection. And so that led to the research that you heard about from Professor Charpentier. In other words, we worked together to determine the function of the protein Cas9. And in this cartoon, the protein is in blue. It holds on to two types of RNA, the CRISPR RNA right here, that contains a sequence that in bacteria would come from a virus that can recognize by base pairing a sequence in DNA molecules. And when that interaction occurs, the Cas9 protein can cleave the two strands of the DNA double helix to make a very precise cut in the DNA at a sequence, a site determined by this part of the CRISPR RNA right here. And then you heard from Emmanuelle that this system also requires a second RNA called tracer that was figured out during our collaboration by the work of Martin Janek in my lab and Krzysztof Chylinski in Emmanuelle's lab. So this is a programmable enzyme requiring two separate RNAs to allow it to find and cut a sequence of DNA. And importantly, this sequence right here of letters in the RNA of the CRISPR molecule is easily changed. So it's possible to do this, make a change in the sequence here that allows this to find and cut essentially any DNA sequence that one might want. And then as you heard from Emmanuelle, in the laboratory, once we understood how that molecular mechanism, it was then possible to link together the two separate RNA molecules that occur in nature to form a single guide version of this RNA that would include the information for binding to Cas9 and the information for targeting DNA. And by doing this, we created a simpler system that could be deployed for a different purpose in other kinds of cells, namely for genome editing. And so that brings me to technology. And so to explain how this works in a little bit more detail, I wanted to show you this 3D printed model of Cas9. So this is a model that sits on my desk in Berkeley. It's about this big or so, and it's based on a crystallographic structure, so a real molecular structure of this complex. And what this shows you is the Cas9 protein, the white molecule, holding on to the guide RNA, which is the orange molecule, and interacting with a blue DNA molecule that has a sequence of letters in the DNA that matches the sequence of letters in the RNA. And so when that occurs in a cell, this protein has the ability to unwind the two strands of the DNA molecule to form an RNA-DNA helix inside the protein. And that, in turn, positions two cutters, two catalytic centers, two chemical centers in the enzyme to generate a blunt double-stranded break in the DNA molecule. So it's truly a programmable system, and this is the key to using this as a technology, is that the same protein can be easily programmed by changing the sequence of letters in the bound RNA, something that anybody with a little bit of training in molecular biology knows how to do. And so to connect that to technology, and you saw this in Emmanuelle's talk, but this is really a slide that just illustrates that although in bacteria, a double-stranded break in the DNA leads to very rapid destruction of that DNA molecule, so that means this is a great system for bacteria to use to destroy virus DNA. In eukaryotic cells, meaning plants and animals and fungi, the cells have a very sophisticated way of fixing double-stranded breaks in the DNA, and they can do that by a couple of different pathways that lead to either a little disruption in the DNA sequence during repair or to integration of a new piece of DNA during the repair process. And so many scientists had, over the years, had appreciated that you could harness these pathways, these natural pathways for DNA repair, if you could figure out how to introduce a DNA double-stranded break at a position where you might want to introduce a change to the DNA inside of a cell. That was the challenge. How do you introduce that double-stranded break where you want it? And that's where this programmable enzyme from bacteria comes in, because that's exactly what it does. You can program it to find and cut DNA to produce this, and then the cell takes over and does this. So I'm just going to show you briefly a second video that is a sort of an artist's imagination of how this might work inside of a plant or animal cell.
Scientists have long imagined being able to make specific changes to the genetic code of a cell or organism. A revolutionary genome editing technology invented by scientists at UC Berkeley and Umea University makes this possible using an RNA-guided protein called Cas9. Cas9 searches through the cell's vast genetic material until it finds a sequence of DNA that matches the sequence of its programmable guide RNA. The guide RNA enables Cas9 to open the DNA helix and to position 2 molecular blades to cut each strand of the DNA double helix. Repair enzymes seal a gap in the broken DNA, sometimes by inserting new genetic information supplied by the cell or the experimenter. In this way, disease-causing mutations can be corrected by changing the underlying genetic code. Scientists are now using Cas9 technology for research applications in human health, agriculture, and bioenergy.
So just to show you a few examples of things that are already being done with the CRISPR-Cas gene editing technology, I wanted to show you some of the headlines in a magazine called Wired magazine that have come out over the last couple of years. And I'll just go through these quickly, but I just want to point out that in agriculture, it's been possible to use gene editing to create hornless cattle, cattle that don't have horns, and therefore in captivity, they don't injure each other with their horns. It's been possible to show that you can, at least in the laboratory, make changes to blood cells that would correct genetic diseases of the blood, like sickle cell disease or thalassemia. It's been possible to use the Cas9 system in insects to create insects that are not infectable by organisms that would potentially lead to infection in humans, like dengue virus, for example, and other kinds of pathogens. It's been possible to use the CRISPR-Cas9 system in animals that have in the past not been genetically tractable. And this is an example of experiments done in Tasmanian devils, these little guys right here, to figure out the genetics of cancer pathways in these animals and how that might compare to what we find in humans. It's also been possible to show that you can use the CRISPR-Cas system in pigs to generate pigs that might be better organ donors for human patients. And there's the potential, and I'll come back to this, to use this system in various kinds of embryos, including human embryos, to make changes to DNA that are not only occurring in the organism itself, but also become inheritable by future generations, something that I think raises powerful sort of ethical challenges. And then over here, we have a headline that points out that there already are ongoing clinical trials, this is one going on in China, to use the CRISPR-Cas9 system in cancer patients to harness the immune system to more effectively find and destroy cancer cells. And then finally, I just want to point out that there's, I think, a lot of incredible research now enabled by this technology in organisms that in the past were genetically intractable. Things like butterflies and various kinds of rodents that, and other kinds of plants that are now being studied in laboratories for research purposes and moving forward the whole field of biology at just an incredibly rapid pace. So finally, just in the last couple of minutes, I want to turn to this question of ethics. And for me, this really has been a fascinating experience because I'm a biochemist and I've always studied molecules. And then through the research that I did with my colleague Emmanuelle Charpentier, we realized that there were some powerful ethical challenges that resulted directly from the work that we had done together and this technology that we had developed. And in particular, this issue that I touched on earlier, and I just want to point out here, is, I think, very profound, which is the ability to do what we call germline editing. And this is an example where you can see a pipette here that's holding a fertilized egg. This is a fertilized mouse egg a needle is coming in from the right and it's injecting the CRISPR-Cas gene editing molecules. Now, when those molecules are injected, they can alter the DNA in this embryo in such a way that the resulting organism has genetic changes to all of its cells, and not only that, it can pass those changes on to its children. And I'm just going to show you one quick example of this, a visual example. I love this example. I got this from a student, Sanada Kodka from the University of, she's actually at MD Anderson and the University of Texas. And it's an experiment she did in frog embryos, and it shows a couple of things. It illustrates germline editing, and it illustrates how easy it is really now to do this. So what you're looking at here are 2-cell embryos. So these are frog embryos. They've divided once, so they have 2 cells. And what Tsunada did was to inject the CRISPR-Cas9 molecules into one cell of these 2 cells in such a way that she removed a gene important for making the brown color of these frogs and inserted a gene that turns the cell green. And so you can see that you have these beautiful embryos where one cell is now green and the other cell is normal. And when these animals develop into tadpoles, you can see that in the top image, you can see right down the center of this animal, the cells on this side are lacking the pigment gene, and the cells on the top are normal. And if you put them under fluorescent light, you can see that all the cells on this side of the animal are now green because they've been edited, and all the cells over here on the other side are not because they're normal, okay? So that just shows you the incredible precision of this kind of technology, and it wasn't difficult to do. Furthermore, you can do this in other kinds of animals. So in early 2014, a paper was published in which scientists had done that kind of germline editing in monkeys. And when I saw that work, I realized that it might already be the case that scientists somewhere in the world were thinking this might be an interesting thing to do in this animal. And this is actually a picture taken from the cover of The Economist magazine from last year, in which it was sort of pointed out that we now have a tool that allows, in principle, changes to the DNA of human beings that could not only correct genetic disease, but in principle could also lead to various kinds of enhancements. And so the question comes up, who decides and who gets access? Should we do this at all? And I think it's a really interesting set of questions that I've now been engaged in discussing with my colleagues. We actually organized the first meeting about this kind of germline editing in humans back in January of 2015, and that has spurred worldwide discussions as well as a series of meetings, international meetings, to discuss this and to help educate people and our governments about this technology and encourage responsible use and responsible regulation. So I'd just like to close with a couple of thoughts. First of all, it's important to point out that the development of this technology was and is a team effort. It includes academic institutions, professors and their students, clinicians, other researchers at other kinds of nonprofit institutions, governments, taxpayers, and companies. So it's become a very interactive kind of thing, and it's been really fun to be part of the development of all of that. My own affiliations are quite varied, and I'm just pointing out here that I'm part of quite a number of nonprofit and academic organizations, and I'm funded by these groups right here, and I'm also part of a number of companies, or advising them, or a co-founder of these, that are involved in developing the CRISPR technology for different kinds of commercial applications. I've also been involved in starting the Innovative Genomics Institute. It's a partnership between UC Berkeley and the University of California, San Francisco, which is our medical school in the Bay Area. And please check us out on the web. We're an educational nonprofit organization. We focus on research and also education and ethical discussions. And that's it. Thank you very much, and it's been a pleasure to be here.